That's interesting.
Oh, he's beautiful.
Looking down at you at 9-0.
Oh, look at those little crinoids.
Let's go full wide.
Going back to the octocoral that we collected and talking about how we identify them, Ira Sampaio from the University of the Azores points out that you really have to look at the what's known about octocorals in the region where you're looking.
Is that new?
It's an interesting sponge.
And in the Azores, there's 2 species of Narella. One is whip-like.
Any interest in this watch lead? This sponge here?
Let's take a look at it.
Okay.
Thanks.
We're hoping that some of the folks in the chat room can give us more information on the skate we just saw, including its gender. Does anyone out there know?
Ship's still moving, right?
Go ahead, Watchley.
Yes, I believe so.
Okay.
Did you call me?
Yeah, it looks like we're still.
Nope, I don't think so.
Oh, sorry about that.
Oh, that's okay. We have a tiny little crinoid on the back of the sponge.
Yeah.
Yeah, Nav, I see what you mean by putting in the temporary
waypoints. Let's come out, video.
That helps a lot.
Go past the skid.
I think we need to cruise up slope for a bit. Our goal is to reach the summit of this mound where we would expect to find the highest densities and greatest diversity of these coral species that we've been seeing here, but also very different communities at the top of the mound structures that we saw yesterday farther to the south. Have we seen any of the Lophelia at this point? Okay.
And we actually haven't seen any live stony corals except for a cup coral or 2.
Well, hopefully as we get up into the current, we'll see both.
And you can see pieces of kind of more recently dead Analopsamia profunda here.
Updated calculation on the time we're going to have on the peak. And after we visit all the waypoints, we'll have about 107 minutes left over.
Great. Okay. That sounds like more than enough.
We have a few more sponges here.
I have you there at 8-0.
Copy.
See a little more relief right in front of us. Might be similar to the type of rocky structure that we saw yesterday. We got a sample of the only consolidated part that we witnessed, and it was a little ledge. It's hard to tell right now if this is similar to that.
And what did you think once we got that sample up?
Yeah, the ledges that we sampled, got a nice piece of it. It was basically cemented together coral rubble, which is what we thought it might be. But it had certainly long since been buried and
let it sit for a minute or
cemented together by some of the materials in the sands and the
So anything of particular interest here, watch lead?
Another interesting coral. I think we saw one of those yesterday. I'm not coral, sponge. Maybe a quick zoom to this sponge real quick.
The
one
in the
right
middle?
in the middle, yeah.
Okay, go ahead and do a quick zoom video. Hold that. Looks similar to the one we saw earlier.
Yeah, I think we saw 1 or 2 of those yesterday as well.
Mm-hmm.
I'm not sure we got an ID on it, but...
Okay, let's come out, full white, minus the skids.
You can see some little pink cup corals in there as well.
So we'll just do a broad survey of the area and we'll keep the ship moving.
Yeah, that sounds good.
Watch lead, Snev. Be advised, we recently passed waypoint 2.
Ivy there at 90.
Roger that. Thanks, Nav.
Copy.
We've passed waypoint 2 now on our dive track.
ROV nav bridge, move complete.
Thank you, Bridge. Thank you, Bridge.
It looks like we're seeing more of the little octocorals that might be similar to the Swiftia that we collected yesterday. So Ken Sulak gave us some more information about that small skate that we saw.
There at 75 degrees.
He identified it as Fenestraha plutonia. And he said that the hind lobes of the pelvic fins are broad and fin-like, so that means that it's a female. And the males have those hind
So you
lobes
sponge
modified
that bright
into
green
club-like claspers. Great, thanks for that, Ken.
So you
So
can
that
come
is
down.
the
Yeah, I was
same
gonna say, if you want me to
one
come
that
down.
we've seen on the Charleston bump, the common name of Pluto skate, or at least that's what we were calling it.
Okay, yeah.
Just a smaller version of it.
That was a tiny
younger.
one.
Yeah, it really was.
See, I'll call out like winch stuff if I need it, but I'll leave it up to your discretion. If you think you need to come down, you can feel free to do so.
Copy that. I've dropped down 3 meters, so you should have some play.
Copy. Thanks. We're gonna go look at this bright yellow sponge real quick.
Sounds good. We're going to take a look at this bright yellow sponge in
Okay,
the center
one
of the
right in
screen
front of
here.
you, or the triangular one behind?
This one right in front of us.
You also see some of those soft corals, Duva, Florida, that we've been seeing for a lot of the dive yesterday and today, and those are the pink,
Okay, go ahead, video.
pink soft corals.
And we collected one at
Hold
some
that.
point, did we not? Maybe not yesterday.
Yes, we did. Yep, we
And
collected one
It
yesterday.
was very deflated when it came up.
Yep.
Did not look anything like the version you're seeing on your screen.
And there's a little crab or shrimp underneath the sponge. Can't quite tell what he is.
I think it's a hermit. Yeah. Is that his
Might be, but
shell on the back?
looks like he's actually within the coral matrix, but I can't tell. Oh yeah. Oh, sorry. Yeah, it's definitely a hermit. I was getting a strange look at it before. And yeah, we have some branching bryozoans pointed out by Megan McCullough.
It's a
Okay.
nice composition.
Come out.
Yeah.
That image.
All right.
It's a lot of life going on in that small scene.
It's interesting how across this landscape you see the organisms
They're starting to come off to my
keeping
port
distance
side.
from one
Do you
another.
want me to
Some of the
follow you
larger
around?
No,
organisms
no, I'm gonna jerk back.
sort of
Go
claiming
ahead,
their
yeah.
space. And it may just be a function of there isn't enough substrate framework that they can attach to.
Looks like we're getting to a flatter area. Totally covered by coral rubble.
Coming up on my camera.
Yeah, the plan for this dive was to go up the steep slope that we've
We can get
completed,
another move in.
and now we're moving across a broader
Copy that.
area that
Over 30
is at
meters.
the
Sure.
sort of a ridge crest
I have you
of
there
this
at
mound.
5 degrees.
And then we will
Copy.
start climbing again up to a depth of about 720 meters. We're currently at 752 meters. So we have some
Bridge, Nav.
This is bridge
distance
over.
to cover
request a ship move.
and some
Awesome.
Range 30
depth
meters.
changes still ahead of us.
Bearing 300 degrees.
We're hoping to get
Speed
high up
decimal
into
2
that.
knots.
Yeah, so now
Current.
that I'm
Okay,
ahead
we'll
of
go
you again, you
30
can come
meters
down
at
on
a
the winch.
bearing
Who lives on top of the
30
mound?
at a speed of decimal 2.
Good copy, Bridge.
we're predicting a very different population of organisms based on what we saw yesterday. Be interesting to see if we're right or if it's completely different to what we think as well. Everything's new to us here.
And that's one of the things we're trying to discern as we put the pieces of this puzzle together. So we are visiting a bunch of sites during this expedition on the Okeanos Explorer, Windows to the Deep. But NOAA Office of Exploration and Research has actually been doing work here on and off for over a decade. As we
ROV nav bridge move initiated.
As the years go by, we have better technology than we did at the beginning, but still, if we want to provide the best science possible to resource managers, we have to collect all the data that's available, including that was gathered earlier and what we're gathering now. And when you put it all together, you'll be able to have the best understanding of what the habitat requirements are and what particular life histories mean for each of these species. So we continually gain a better understanding of the biodiversity and how it's organized,
that they're always off, but science
and how it's distributed geographically.
Try to compromise.
And how this organization relates to all the environmental parameters that we can measure, such as the temperature, salinity, oxygen, and depth, and then the topography and slope, and the information that we get from mapping. And so each one of these dives allows us to learn more about each species and how all of these environmental parameters affect it. And each dive also tests the habitat suitability models that are a big help to us in trying to discern where corals might be, coral and sponge habitat might be. So we see whether certain species are present or absent at particular sites, and all of this will instruct the coral habitat suitability models, allowing in the long run for more precision in future versions of the model and better predictions about where these habitats might be. Again, this is a very vast area that we're working in the southeast U.S. coastal region here, or not coastal, but deep waters. And we can't be everywhere, but what we can do is visit just a handful of sites and then try to predict what the rest of them will look like. So moving from the south part of Stetson Mesa up to the north, and comparing and contrasting what we see as we move up and over these mounds. All of this is really important information that we might be able to generalize to the broader area.
And what we're doing with these dives, we refer to as doing the ground truth for the data, for the predictions. And in order to even select a site, we would obviously want to select a site that we would predict habitat occurs. So to make those predictions, we first have to get high-resolution maps, which are not always available. In fact, this southeast U.S. continental margin, which extends hundreds of kilometers offshore in this part of the world, has very little of it covered with high-resolution maps. The map that you might look at on the Google Earth image is very low resolution, where each pixel might be 100 kilometers. So any of these small features, such as the mound we are diving on right now, would not even show up on that kind of resolution. It would be like having a camera, an old pixelated digital camera, one of the early versions, and now we have really high resolution cameras with very fine resolution to them. So once we have better maps, then we can see features we did not know even existed, and this mesa is one example. The Okeanos mapped it in detail in 2014 on one of the expeditions, and revealed hundreds of these mound-like mound features. They're not mound-like, they are mounds. And they're scattered across the feature called the Stetson Mesa. And yesterday's dive was, I don't know, 50 kilometers south of here. I'm just guessing, I'm not sure, but south of here on one of the mounds, and we're seeing very similar habitats here on similar features. So if we were to look at maybe 10 of these mounds across Stetson Mesa, then we could enter that information into predictive models and say, when we see this type of feature with this type of return of the acoustic
There's a rat
signal
tail there, though.
would tend to reflect differently on this
Yep.
seafloor instead of bouncing off of very hard rock, which would give a strong signal return. We call that backscatter intensity. We can use backscatter intensity and what the bathymetry, the water depth information,
ROV nav, bridge, move
tells
complete.
us to be able to pinpoint areas that
Bridge.
would be conducive to coral growth. And that's kind of information that gets fed into
shipment
these
just
predictive
completed
models that
Copy.
get
Copy
better
that.
and better
Yes,
as
put
our
another one in.
technology
Yeah,
and as
just
our
roll
information
yeah, just
increases. But
copy
without
that.
ground
Keep going
truthing
uphill.
like what we are doing today, we would not know. We could only make hypotheses as to what the seafloor has growing on it in terms of the biota. And also in terms of what the substrate character is.
Bridge, Nav.
This bridge over.
And I think
Like
that
something up
ship
this
move?
type of research,
Okay.
given this
30
very
meters,
broad area that we're
bearing
trying
of
to
300
characterize,
degrees.
just
Speed
Plenty
decimal.
of
calls
tether
for
if you wanna-
collaborative efforts.
30
So
meters, bearing
Yeah, plenty of
30.
tether if you
USGS
wanna-
has done a lot of
Over there.
mapping.
Bridge.
NOAA has done a lot of mapping. There are different
I'll
resource
actually come down a little bit more
managers
so you get a little bit more.
who seek similar types of information. And when we can have all of these agencies working together,
That's fine,
we can
because,
bring the
keep
best
an eye-
science
you look at your
available
sonar right
to
now, you're
resource
within that
resource
12.5 meter
managers.
circle there.
Yep.
So general rule, you don't want to get any closer than that.
Copy that.
And just a few minutes ago, I commented on how the organisms were spaced across the landscape more. Here we're seeing them more clumped into these
Oh, that's some
frameworks
interesting
that
fish
are a
there,
little
too.
more mounded.
Oh,
Oh,
we
is
now
that a
bridge
black
move
belly
initiated.
rosefish?
Right
Thank you,
behind it.
Bridge.
Go ahead and zoom in
Could
video.
be.
It's the right color. Or
there's-
maybe it's a
Hold that.
a red-eye gaper. It's one of the
No, I-
Chaunax.
No, I don't think so. I think it might be a scorpion fish.
Oh, cool.
It's either
It has the big lips.
yeah.
That
So I
should be
thought it
closer.
might be one of the gapers. But you're right, the fins aren't modified for
Yeah.
walking.
There's
Push
a
in
long
and try to
spine
get a little
scorpion
closer.
fish that's found in this area.
Watch leads, Nav.
So, yeah, you
a
can
few
definitely
minutes
come up a
ago,
little
we
bit
passed
now.
200 meters over the bottom.
Roger that, Nav. So we've traveled 200 meters since we've reached the seafloor now on our horizontal transect up this complex feature.
Here, go
Oh,
ahead
and maybe
dig
that's our
lasers off.
first live
Okay, go ahead.
sclerotinian
And you
there
can
on the
come up
left.
on the winch a few meters as well.
Coming off.
Yeah.
So I can set down light enough that he won't get scared away. Okay, go ahead, video.
He doesn't look afraid at all.
Hold that.
It does seem as though he has a modified fin, though, underneath the other.
Do we have somebody joining us on the line?
We're hoping it'll be a fish expert.
Looking down at you at
Tighter.
6-0.
After you look at the fish, if you would look in the lower right corner, there's no hurry, but there's an octocoral there that might be a branching primnoid that I'm not sure we've seen.
Okay, Scott, we'll take a look at that. After we look at this grumpy fish.
Yeah, it's beautiful coloration.
Yeah.
Get a good footing here. Well, let's come out. I heard an octocoral to the bottom right. Is that correct, watch lead?
Yes, that's correct. Thank you, pilot.
And you're going to have to help me out on this one.
ROV there at 7.5.
Copy. Which coral is it?
It was a special request, so I'm not actually
Let's go
totally
partial
sure.
video.
It might be just a little bit to the right, that slightly less white coral. Is that correct, Scott? Oh, or are you talking about the one that's larger behind that?
No, the one that's almost just past the end of the tail of the fish, kind
Okay.
of wispy branching.
That
Okay.
one up
Yeah, so
there.
Pilot, it's the one that's behind the tail of the fish, the wispy,
Okay.
taller one.
Yeah, go ahead and video. Zoom in. Hold that, please. So that one
Kind
there.
of, grayish white from my perspective.
Right.
Yep, that's the one.
Looks like there's...
Trim know it for sure.
Okay.
Looking down at 9.0.
Beyond that, I'm
Zoom in
not
further.
sure exactly what species, and perhaps the one that's on its right is the same thing. Maybe two colonies of the same thing.
Yeah, I would guess
How
that
much
those are
left
the
in the move?
same.
Another 15 meters or something.
Just
It
about
looks like,
done.
is that
Oh,
an
okay,
anemone
great.
on it?
So,
Yeah, it does look like there's an anemone on there. Looking at it from the back side, the pedal disc seems
Another
to be
5,
wrapped
6
around
meters.
the
Oh,
branch.
perfect.
Right.
So this may be another species in the genus Candidella. I'm looking at the polyps, and the polyps are standing almost perpendicular to the branch, and it's one of the clues that we use. Are they perpendicular? Are they pointed downwards or upwards? Things like that. Thank you for the close-up.
Great. Thanks, Scott. And thank you, pilots.
Okay, we can come out video. Go full wide video.
pilots and watch leads, be advised at our current pace, we should have a little bit over 100 minutes of time on the peak. That'll be after we visit waypoint 5.
Roger that. Thank you, Nav.
ROV nav bridge move complete.
Thank you, Bridge.
Wait, that will be after we visit waypoint 5?
That's a good copy, co-pilot.
Wow. Should we try and do like a lawnmower pattern then, in between 4 and 5?
Wow, it looks like we've got more standing dead coral coming up here as we
Yeah, we
head
can
up slope.
just look around.
and
instead of
many
just
more
going straight to
sponges.
waypoint 5 and then
Yeah.
kind
backtracking.
of leaning back in the current.
Yeah.
Probably be a lot more stuff up there. We can
Definitely.
take more time, do more shots.
You can see the deposits of sediment behind these in the lee of it. And on the bottom of the screen, just about to go out, is also a pancake urchin. We saw several of those earlier in the dive. Haven't seen them for a little while. And Cheryl, you mentioned that you were starting to see the scleractinian corals.
Yeah, I wonder if we
It's
can,
another sea star over on the left.
when we get another glimpse of one, we can stop and look more closely at it. Try to determine what species it is, but wow, we're going uphill in a big way now. Pilot, could we get a closer look at the white coral right left of center?
Yep.
Thank you.
See the current whipping by those small corals on the left.
Yeah, the current is really strong here. You can see all the soft corals blowing in the current. Yeah, this is looking like an Alapsamia to
Ship
me.
stopped, right? You said. I think I heard the bridge call down.
That's a good copy, pilot.
Okay.
We're stationary.
Okay. Okay, go ahead and zoom in video. That's the one,
Yep.
right?
Yeah, that's great. Yeah, that looks like an Alapsamia to me.
Go in tighter.
And one with
Copilot.
living polyps. We've seen a
Yeah.
lot of this today,
Copilot,
but not living. In fact, most
another
of the
advantage
rubble
of
that
using the
we have
markers
crossed
for
is
travel
likely
on iPad
made of this species.
Is it's easy to see
Skeletal
when the ship
material.
drifts.
And we zoomed in a couple other times
whenever
and
we're
didn't find
stationary, a
any
lot of times I
live
like to make a
polyps. Or
marker.
did we find
That way
any
you can
yet?
tell, when, how far
We might
away.
have seen a branch or two, but we haven't actually zoomed in on anything
Mhm.
alive yet. So,
Mhm.
yeah, this is the first one.
Some of them just drifted
But
by,
lower on the slope.
Serious as it
Did we find any?
can.
No.
Polyps,
Maybe they're
yeah.
at
No.
90.
So,
Sure.
Yeah, we
Tilt
saw
down,
plenty
I
of
think
the
it goes back to
skeletal
101.
material.
Okay, coming down a little bit further.
The ship stopped a few
Looked
minutes
like
ago,
it might
so
be
Claw
a
down here.
yeah, a
Further.
little hermit crab or
Looking
some
at
kind
you
of
at
Galatheiid crab
100.
that's on the backside of this
Ooh.
coral.
And a couple brittle stars.
wrong?
They're
Just a high pitch
orange,
noise.
Oh.
orangish red legs.
I did not hear that.
Let's see,
luckily.
wrapped
Do
around
you
on
have
the right.
survey one?
No.
Yeah, that's.
I did, but I wasn't sure I heard it.
Okay,
through
Lots
let's
of
come out
current
video.
here.
Let's come full wide video.
And that scorpion fish that we were looking at a little while ago, Ken Sulak gave us an ID, a tentative ID, as Trachyscorpia cristulata. Well, that's exciting. Hopefully his mood will improve as the day goes on. He wasn't looking too happy. You can see more kind of high-standing dead skeletal framework from corals. And I am going to step away for just a minute and leave Leslie to guide this.
Pilot, can we take a quick look? Are you able to
Yeah,
stop
absolutely.
at the sponge on the left?
Yep. This one right on the left side of the screen.
Yes,
The yellowish
that's it.
one.
Yeah, the bulky one
Yep.
that's almost center now.
Okay.
Yeah, I'll be back there at 90.
Copy.
And I'm seeing that Tina Molodtsova from Moscow has been tuned in every day and very helpful on the
lasers
chat
off.
room. And she's
Lasers
Go ahead
coming
and zoom
up.
in video.
commented that we have not seen Anthomastus and black corals today, and I agree. The Anthomastus, they're very easy to spot. They're that bright red, pinkish. We haven't seen those, and I don't know if we've seen any black corals. I don't recall seeing any, but I've been away from the microphone at times. But that's an important observation. We did see a lot of both of those groups yesterday. And we're looking at a sponge I don't think that we have seen today. And Tina is from the Shirshov Institute of Oceanology in Moscow.
Okay, is that enough of a view for you?
Yes.
Okay, we
I'm
can
not
come out
getting
video.
any feedback from the sponge people on the chat room, so we'll keep moving on.
All right.
And again, you can see how the larger organisms are spaced
Rat tails. Or
from one another rather than in clusters. And Ken Sulak contributed earlier on the chat room that this was likely an indicator that there are limited resources, so they pretty much find their own ground so they can claim their own particulates that might be floating by. Less competition.
I have you there at 7-0.
Okay.
Where we see the clusters is where we see any kind of topography from
You can get another move.
this dead coral framework that
Copy that.
creates
Another
these
30
smaller
meters.
mounds
Sure.
on top of the
30.
large-scale mound that we are crossing. In the foreground, maybe we can take a quick zoom on that.
Which one?
The pancake urchin.
Sure.
We've seen several of these, but I'm sure many viewers have not gotten a close-up look.
I'm going to get a little closer. Yeah, you can come down a few meters, Lars.
Yeah, I'm working my way
Bridge,
down now.
Neff.
Okay.
Bridge over.
Like to request a ship move. Range.
These urchins
30
are in the
meters.
phylum Echinodermata.
Bearing 300 degrees.
We've seen many of their relatives
decimal
in
2
different
knots.
classes. The
Okay,
sea
we will go
stars, the
30 meters at a
Asteroidea.
bearing of 300, speed decimal 2.
These are
Thank you, Rich.
I
Echinoidea.
have you there, pilot,
Class
at
Echinoidea.
6-0.
Okay, go ahead and zoom in video.
We've seen pencil urchins, which are also in the same class. And here you see
Hold
the
that.
tube feet that this one doesn't have as many of the modified tips of the move feet. I guess they do have a little bit of
Going
extra
tighter?
tip to it, and those are
Go tighter
sacs on
video.
the end that I believe have a venom associated with them, so they're no fun to pick up. We don't intend to. But they're also inflated with water, and when they do come up to the surface, they are flattened, which is where they get their name, the pancake urchin. If you've seen sea
ROV
urchins
nav bridge,
near
move
shore, they
initiated.
look more like pin cushions and
Thank you,
have
Rich.
a calcium carbonate shell to them. And those spines are much more solid, the spines on the shallower urchins. But we also do see the pencil urchins in this area, as I mentioned, and they have very thick spines. In fact, right at the base, you see a couple tubes that are brown from the iron oxides.
Is that good enough for your purposes?
Yeah, that's great. Thanks, pilot. And those brown
Okay, let's come out
tubes
video.
look like pencil urchin spines. And in the rock record, that's often all we ever find of urchins is their preserved spines. You can imagine that that pancake urchin would be very hard to preserve in the fossil record. It doesn't really have any hard part remains. And here's another of the sponges we saw several of earlier. Can we do a quick zoom there? We don't need to stop for long.
Quick zoom. And hold that.
Yeah, and it's almost as though they build themselves a mound and to reach up into that current. That's good, thanks.
Yep. I'll pan over to this other sponge.
Yeah, it gives you a sense that this sea floor here is really almost cemented
Yeah, you
with
come down a little bit
the
more, Lars.
What was wiggling around on the top of that sponge?
Sorry, Scott, I couldn't hear you on that.
Sorry, I was asking you maybe past it, but what was wiggling on the top of that sponge?
Oh, I'm sorry. We didn't do we're doing a quick sort of flyby zoom. Sorry.
Okay, let's come out.
We can go back to it. Pilot, can we just go back to that previous sponge? We have a question on it.
Sure. Get lasers on.
Lasers coming on.
I saw something wiggling, and we were looking at that one that had the
Lasers
extended
are on.
chimney on it, and I wondered, could it be that flexible? I wouldn't think so, but
Scott, what kind of delay do you have today?
Let's go
Are you
ahead
still at
and
about
zoom in video.
7 seconds?
about 8 seconds. I see you're going in on it.
Okay.
What are we looking at here, watch lead? Hold
At the
that.
top, the organism that is waving right at the top.
Oh, interesting. I see it. Let's come out and I'll sit down for a stable view.
Yeah, interesting. So I still couldn't tell if that was actually part of the sponge or something else on the sponge.
Mm-hmm. And we're going to sit down and get a really good look at it in a second.
Lasers off.
Okay. Let's go in video.
And you can see the effect of the Gulf Stream. We are east of the axis of it, but oh, look at that. Looks like a
Over there.
worm that has
6'5".
attached
Copy.
sponge spicules to it.
Oh, that is really neat.
Definitely taking advantage of the available materials.
Yeah, that's quite fascinating. Really interesting coil. So it's not an osculum or X-current opening of the sponge, although it does look like, as
Actually,
you say,
do you want to hit
something
joy lock on
has
the pilot
incorporated
box,
parts
Lars?
of the sponge spicules
On my box.
probably
It's
from
the
an
rightmost
apology
button
worm.
at the bottom.
Thanks for that, Jim.
Joy lock, yep. You gotta
Sure.
hold it down for
Yeah,
a couple
we
seconds.
yesterday we looked at
Thank you.
some coral rubble, and it had an agglutinated foram growing on it as epifauna. I just learned that term. And the agglutinated forams are single-celled organisms, and they grab the available sediment to build their shell. And it was comprised, the shell was comprised mostly of sponge spicules. So a single-celled organism was taking advantage of the spicules of the area.
Yeah, and as you noted, there are a variety of worms that do similar sorts of things. They're capable of building tubes, and then they'll glue various things that are around in the environment to the outside of the tube, I presume for some sort of disguise.
Oh, yeah.
And Tina
ROV
Molotova
nav.
is suggesting a
This is the
sabalid.
bridge.
Go ahead, Bridge.
That move is
And
complete.
Shirley
and I'd like to bring
Pomponi
that
is agreeing,
About 3 degrees to starboard
and
to
that
a
it
heading
is
of
not
200.
the oscular tube.
Copy. This is a good time to do that.
Okay, thank you.
Thanks. So ship move is complete. They're going to take a few minutes to change their heading.
All right, well, let's zoom out
So
and
you got a little bit of time.
Go
head
ahead,
on,
video.
unless we see something else. Unless there's
So
any other
Nav,
comments
were you
from
flying
the
with
chat
auto heading,
room?
or you just put it on?
We
for
have
live
I just
on?
put it on. to hold you.
36
Okay.
people on the chat
Swap.
room right now, so
Okay.
appreciate all your comments. Sometimes it's hard to keep up with them. I'm flying solo now, but I'll do my best. All right.
I have you there at 90.
Copy that, Copilot.
On our first biological collection, we
So I've
used a
tended to
scoop,
kind of
so we were able to
face
get
the
some of
direction
the
that you were just
sediment
in. So
Okay.
face kind of
on this seafloor, which
southwest,
is
because
very
that's-
nice
then you
for
have
me
the
to be
current
able to
coming right at your nose.
see what
Copy
makes
that.
up
And it's easier to
this
get a stable
particular
shot.
region sediment.
Easier to fly.
We were farther
and then
out
you don't dust up the
on the
shot when you
continental
set down.
margin, well
Okay.
So
offshore
you just
from
kind of
here,
saddle along looking in
and
that
beyond the
direction.
Gulf Stream's
Okay.
effect in areas that were deep enough, 1,300 meters to 3,300 meters. And those areas
Wow.
were being affected by a very different current, the western boundary undercurrent.
ROV nav bridge,
And
we are
much
spinning
colder
up on a heading
current,
of
slower.
200.
A
Copy,
lot
Bridge,
slower
thanks.
than the Gulf Stream. And the sediments there were much, much finer because of that lower velocity. Here we have relatively raging current, which does not allow the fine particles to settle out. So here we see in the sediment, what I'm predicting we'll see is large foraminiferous shells, those single-celled organisms that live in the sea surface.
So you're doing a
and,
lot of
pteropod
translations
shells
now.
that are
Yeah.
swimming
Just kinda-
gastropods.
So, and I was trying to
and
tend
other
to stay towards
organisms
the
that
center or
live
right
in
side of the
surface
screen,
waters,
just
with
so
an
then if you
occasional
see something ahead of you, you
seafloor
can
dweller.
get to it.
There
So right
are
now, if you
many,
see something
many
ahead of you, you don't have enough time
thousands
to get
of
there.
species of benthic foraminifera, but they are less common in a sediment sample than the
I'll just let
planktonic
it push me
forams.
back a little bit.
Perfect.
Video, pilot, could we zoom in? Almost dead center is a sea star.
Okay, I see it right in the middle.
We get lasers on.
Lasers coming on.
Here, I'll sit down. So
A
you
little
think-
one, but we're definitely seeing a few more sea stars up here than we saw at the beginning of the dive.
Okay, watch lead. I'm just working my way over there now.
Excellent, thank
Do
you.
you want a quick zoom or do you want a close inspection?
I think a close inspection. We can
Okay.
also get a look at the sediments surrounding.
We have
Since
time.
I was just
so.
talking about them. That coiled organism
Okay,
that
come on
had
in,
the
video.
sponge spicules that we were just observing about 3 minutes ago, Shirley Pomponi says,
I'm gonna
might
rotate
actually qualify as the weirdest animal of the day. This one isn't quite so weird. This is a sea star. I certainly don't know the group or the genus or anything, and I don't know if any of our listeners or chat room folks, hopefully Chris Ma will chime in. He is our chat room specialist on the sea stars. Most of the sea stars have this pentameral radial symmetry, the 5-pointed. And they all have a water vascular system that pumps through the little tube feet, allowing it to mobilize itself, use the tube feet to move across the seafloor. Many of them are scavengers, many of them are predators. You can see the eye stalks at the ends of each of the rays. And they have a great ability to regenerate their limbs. This one has an interesting little polyp on its limb that is at the top. I don't know what that is. Might be a tumor.
So are you using most of your verts to stay on the bottom, Nav?
Yeah.
Okay, pilot, let's
Yep.
go mosey on.
Great. Thank you, watch lead. All right, full wide video.
Hello,
Hi,
It's Chris
who do
Ma.
we have
Yeah,
calling
it
in?
was
Is this
taking
Chris?
quite a bit
this
to
is Chris Ma.
Hello, Chris. We
stay on
sent out the
bottom
bat
today.
star
There's
for you.
a lot of current.
Yeah. Thank you for that
It's kind
delightful
Tending to want to push you
little
up.
introduction
water
to sea stars as you
So you can see our Z-Vise
were
is set pretty heavy today.
undoubtedly stalling for time.
Copy.
Well, I
Yes.
didn't
Okay,
How did you
co-pilot, I'm
know?
going to try and get
time it
Out
to
in
exactly so X
Copy
number of
that. I
seconds
have you at
to
90
see
right
how
now.
long I would
Copy, thank
it
you.
would take for me to dial in. So the sea star that you guys have focused on is, I believe, in the genus Henricia. And this is a globally distributed genus of sea stars, which terrifies almost any starfish taxonomist, not because they're so distinctive, but because they're all the species are so closely look
I'm
so
not sure,
closely
but
alike.
there's another one
Many of them
That-
are
that light
ecologically
is
important. They feed on sometimes
on the
sponges
screen.
and other
Yeah.
kinds
Yeah.
of
It's giving me a
sort
wicked
of
migraine.
epizoites, so small microfilm, other kinds of non-moving prey.
Yeah, I don't know. It's
they're
going to be going for a while.
they're
Do you want to
widely. They can be quite abundant
You
where
wanna
they occur.
swap
They're sometimes
Nav
found in
and
shallow
co-pilot for a
water,
bit?
but also in deeper water, down to I think
That's fine.
about 1,000 or so meters. And in areas like this, they're not entirely uncommon. They sometimes are feeding on the sort of little gooey organic material that's living on the surface, or perhaps there are sponges that are out of sight. Some of them actually have a hyaline tooth that occurs around the mouth, which, although it's never
Right there,
been
5'8".
shown to
5'8".
be actively
Copy.
used in predation on sponges, is often associated with species that feed on sponges. Unfortunately, getting anything beyond a genus on an animal like that from a picture, even a very good one, is extremely difficult. Looking at a specimen and getting a species is often very difficult. So, there are sometimes very distinctive ones that can be picked out of a row, but unfortunately that one will forever remain a mystery, but one never knows whether
And 5-0.
it might have been new or
Copy,
it
5-0.
was simply one that was known. but
Yeah, you can come
definitely
down to like
ecologically
15 meter
important
delta
component.
now.
Making my way down.
And in the
Okay.
Pacific, we saw any number of new observations of them feeding
So I think
on
my,
sponges.
I guess I was trying to
there were some that
quantify
were quite large, feeding on,
how I do that,
really
and
enormous glass sponges. And
as I guess
those
between
were all
30 and 40
valuable
meters,
observations,
that would be
such
like
as,
10 meter delta.
and this one is as well. I'd also add that of the two Goni
Or, sorry,
asteroids
30 to
that
40
you
degrees,
have
I would be at a
been
10 meter
seeing,
delta. And
the
like
yellow
40
sort
to
of cookie
80
star, of
degrees,
course,
I'd be about 15.
is one that
And
I
then 80 to
identified.
90
But
to
the other
100,
one,
I would be
the
20,
other sort of
or 18 or
orangish
20.
one, that has
So that's
almost kind of a
general.
prickly surface.
Looking at you.
That was very interesting because
Copy
it
that,
doesn't look like
40.
one that I immediately recognize. It might be in the genus Cladaster, or it might be in another obscure genus
So
known
those
as
kind of
Floriaster.
like push me over
I'm not
to the
sure
right.
yet, but
Okay, ready for another move?
it's definitely
I
worth
am.
a closer
All right,
look
we're
the
gonna
next
do
time
30
we come
meters,
upon one.
300.
Those were, I don't know how big they were, but you had come upon
Bridge,
one
this is
earlier.
RV
Bridge over.
Another move,
perched
please.
on a stylaster
Another 30
coral.
meters, that's 30 meters, bearing 300
That would
at
also
0
be a
decimal
novel
2 knots.
observation,
Okay, 30
especially if it
meters
turns
at a
out to
bearing
be
of
new.
300 at
So,
a speed of decimal 2.
So
That's
things
correct.
remain to be
Thanks.
discovered, and I look forward to the next happenstance where you guys come upon a sea star.
And Chris, again, what was the species you thought that was? You said Henricia,
well, the
but
genus is called Henricia.
uh-huh,
Henricia.
right. I thought you had also
Copy
suggested
that,
a
Copilot.
species name.
Yeah,
No,
definitely.
no. In fact,
Oh,
I was-
because we.
Seems like here it's
ROV
pushing me
nav
down.
bridge, move
Okay.
initiative.
Copy bridge,
that-
thanks. What's up,
that
Andy?
because
It
these are
said right here,
so
it feels like
difficult,
it's pushing me
these
down a
are
little
similar to one another,
Interesting.
it's often difficult to get a species even with a specimen in hand.
Yeah,
Okay.
now, do you mind
The
switching?
reason I
Absolutely.
ask is
That's
the
fine.
Charleston Bump Expedition, which is just north of here, still beneath the effect of the Gulf Stream. We collected a specimen, and it was identified as Henricia downii.
Mhm.
And it looked similar, but I couldn't tell the difference. I do have several photographs of it.
They all look similar. That's the problem.
Yeah, yeah. Okay,
Yeah,
well,
I mean,
thanks.
It could be. I mean, it's again. That's the problem. Is that that's the reason why they can be so formidable. Is that even with a specimen in hand,
All right.
they often look very similar to one another.
Mm-hmm. Okay, well,
So
great.
even though there was one called Downii, named for Smithsonian echinoderm marine downy, by the way.
You all right, Lars?
Oh, really?
Yes. Even though there is one identified to species, it could be the same species around here, but there are multiple species of Henricia in this area. So, it's difficult to
Just
say.
looking over
especially
the other
from
way,
a picture.
co-pilot.
Copy.
Well, if we collect one and find that it's a new species, we will name it Mahii. After
Well,
you.
It would be, someone else would have to do it then.
I realize that's why
Certainly
we would have to do it.
Yeah,
But you've been a
think
tremendous
you guys
help.
I would much rather
So we
find
get
that
too much
Goni
further
asteroid and
ahead.
name it after
We're
all
at
of
the end
you.
of
So
your tether.
All
Copy that.
leave it.
All right,
Oh,
well,
yeah,
we'll
but if
send
you're
the
right,
Bat
come
Star
drift
out
back
again if
and
we see
come
another
back
one.
with Sirius and go lateral
Very
a
good.
bit.
Thank you very much.
Thanks
Have a good
again,
day.
Chris.
Bye-bye.
And again, that was Chris Ma from the Smithsonian
But the ship is moving, so be up that way momentarily.
and a colleague of one of the scientists on board who was our sampling manager, Stephanie Bush. You've heard her voice periodically. She's also the main voice, the primary voice on the midwater dives that we have done. I guess we've done 2 of those. We'll do a couple more in a few days. Right in the middle of the screen, another of the pancake urchins.
Do we
Pilot,
wanna
we
take-
can continue to
Oh,
head up.
We're waiting for Sirius to
uphill.
catch up right now. But
We're
we're
currently at a depth of about
moving.
247 meters. This is about the depth of the 3rd mound we visited yesterday. This portion of the mound has been a little more barren of high density clusters. Here's a little ridge-like mound And you can see very well how the organisms
Still drifting
cluster at
back
the
a little
highest
bit,
point of
co-pilot.
any
Copy.
small topographic feature.
Take a look at this sponge and corals over here.
Coming up, tilting down, looking at you 45 degrees.
Copy.
Regarding the sea star that we had seen earlier that Chris Ma was talking about, that genus, Henricia, he said was the terrifying genus, and Tina Molodtsova mentioned that she had a grad student who worked with that genus, and she abandoned them immediately as she graduated. So if anyone out there is listening and wants a challenge in the echinoderm world, we have a perfect asteroid genus for you. Yesterday we saw a lot more of the bamboo coral and some really large ones. They were beautiful. In fact, Scott France, the one on the chat line who is particularly biased towards the bamboo corals, professed that it was the king of the mountain, king of the mound yesterday. I don't know who would claim that status in this particular dive. We haven't really seen anyone
Copy that.
dominating
Good orientation right
species
there,
that I can tell, unless anyone would like to suggest or nominate a species.
In my defense, Leslie, yesterday, when
Just
I
gotta
said that,
kinda
we were
move
looking
forward
at a
along
coral
my current
mound,
heading,
and quite literally,
drift
the
over
bamboo
back
corals
into
were
the middle.
growing on the very
Copy.
top of it.
Sounds
Yeah.
like a good plan.
You don't have to defend yourself. We agreed.
Awesome, thanks.
It's looking good. It's
I
very
thought it
smooth.
was hands
Thank
down.
But it is an interesting observation that you make. You know, we saw many of them yesterday. We were in a very similar
ROV nav bridge. move complete.
environment, similar habitat. The depth
Good
isn't
copy,
that much
Bridge. Thank
different. We're only a little bit further north, and yet we're missing both of the species. We're seeing those very large keratoisis, and we're seeing the really wispy cladorisis, and I haven't seen any of them today.
And as Cheryl mentioned, we are seeing few of the stony corals, the scleractinians, that we saw quite abundantly. Different species at the base of yesterday's mounds or in the lower mound areas, and then the Lophelia at the peaks. So we're still hoping to find them as we rise upward.
And there's certainly a lot of coral rubble here, which is an indication that they have lived at some point.
Mm-hmm.
We were having the discussions yesterday about the many factors that have to come together to have some of these vibrant communities, and we're still trying to understand that, which is why all these observations that we make on these dives are really valuable. It gives us one more piece of the puzzle to put together so we can
Right,
better
Alex, that
understand
last move we
where
just
there
ended.
are or not
We're
the
looking to do
most
another one.
diverse and
Keep
vibrant
moving on up the
communities.
hill.
Oh,
Yeah,
we'll
I can't
slow
help
down a
but
little bit.
think that
Copy that.
the Gulf
I think
Stream's
we're making
position
really good time. So
on a daily basis has a great influence. Just when we began our expedition, the axis of the Gulf Stream
noted in the
was in a
log,
position where we
Nav,
couldn't make dives.
how much time we
And
should have
it
on the top.
moves daily, and so we just have to look ahead to the next
couple
day. You
entries
can't
back.
predict it a week out. We're finding.
Might, I'm
And
not sure how long ago
certainly
that was, but
the
you might just
axis
double
is
check.
so much stronger
kind of
than the
doing it
flanks or the outer
every 30,
portions of the
40
Gulf
minutes,
Stream.
just to
And I'm
make
just
sure that we're
wondering
still
if
on track.
some of these
Copy that.
mounds are
We'll,
more
do some
often in the
calculations.
axis or more often outside, and that itself has the greater influence rather than the water depth or the fact that it's on this mesa, and just instead of just thinking it's influenced by the Gulf Stream, it is dominated by where the Gulf Stream's position is on an average, maybe annual basis. And where we're seeing these huge areas of dead coral, perhaps that's where the axis used to be concentrated. Because we certainly see a lot of fluctuation in the Gulf Stream's location and most likely its intensity, its velocity through history. And I'm talking about geologic history.
Cheryl's back. What did I miss?
Well,
Okay.
we had Chris Ma come on and talk about Henricia. Which was a new genus for today,
Oh,
a
wow.
sea star,
300.
and a very common one and a very scary one because it's so hard to identify the different species.
Oh, wow. We had the right expert on board.
Yes.
From afar, anyway.
From afar.
Speed 0.2,
And we
please.
saw some,
Okay, we will move
one of the
30
most
meters
interesting
and
organisms of the
heading
day.
bearing
We
300
saw
and
a
speed
spiral
decimal 2.
tube-like
Bridge.
organism sitting on top of a
Pilots that
sponge,
move is
and
in.
it was completely
Copy that.
coated with sponge spicules that
Oh.
it had used to defend and hide and camouflage itself,
Wow.
and maybe even serve as a way to capture particles.
Those spicules
ROV
are
nav
used by a
bridge
lot of other
initiated.
organisms, aren't they?
Good copy,
Yes, they
Bridge.
are.
Thank you.
Discarded weapons by the sponges get picked up by others.
Yep. And we're seeing a few more of the pancake urchins again. Not in this view, but to answer your question.
Yeah, I just saw one go by,
Yeah.
upper left. And I see we still have plenty of coral rubble.
We certainly do. Not too many clusters. Not the big accumulations or concentrations of the corals and sponges like we saw yesterday near these depths, the higher parts of the
Tilting
mound.
up. 35 degrees.
Copy. Okay, I'll kind of slow down.
Sounds great.
Go ahead, watch one.
Cheryl was looking at our dive track as we are making it and made the observation that we might be actually on the lee side of the crest of this ridge part of the mound, so we might
Nav, do you want
wander
to
over
take a look
to
at that?
the other side.
Did you hear
where
that,
the
Nav?
current would be more
Yeah, I did.
facing, we'd be right on the
Are we looking
slope
to
facing the current.
move
I mean,
to
I
the
think we have
west?
time for
I
that.
feel like the position we're in right now is due to safety of the vehicles, correct?
That'll be
yeah,
a nice
I
contrast
think
to look
maybe
at
what she means is maybe we could
the seaward and
maybe even just
leeward
a tether length over
side.
to the port side on D2.
Copy that.
Okay.
That might just get us on the other side of this ridge because it's kind of wide, right? So we're on the north side, but if we're on the south side, that should be fine because we're already at the top of the ridge, so it's safe. We just want to tend toward the south.
Seeing a
southern
few
border
more
of it.
wispy
Copy
corals
that.
down there, so I wonder if we're starting
Adjust
to
the
see
heading
some
then
bamboo
for the
corals
current
show up.
move, or call it a new move?
No,
or-
we'll let this one finish out, and
Okay, after
we
that.
might let watch lead know that we're 60 meters from the top, something like that. 60,
It was like 117
to
until.
this contour, not waypoint 4, but the contour here.
Gotcha. That one?
The next one.
Yeah.
Yeah, 60 meters to the plateau there.
So by the time we wrap up this move and probably the next move I can do will get us up there.
Yeah.
It'll bring us over to the west side.
Right. So yeah, I would say you can see there's like more sponges and stuff on that side. So when you're down here, just move over to that side of the screen.
Okay. So just start heading over, just moving forward right now. Forward, D2's forward.
Yeah.
Okay.
So pilots, from what I have calculated, and I did a little bit different than what Andy did, once we hit waypoint 4, we'll have slightly under 2 hours to make our way across to waypoint 5.
How did you calculate it?
The time it would take us to get to waypoint 4 from where we currently are, and
Okay.
then subtract that from the total dive time left.
Okay.
And then, so I think what you did was calculate to get to waypoint 5 as well,
Yeah.
and then come back. And what I'm kind of saying is once we hit waypoint 4, we're technically on the top of this feature, and then we can do kind of lawnmower pattern back and forth. We have 2 hours to explore the top, basically.
That sounds good.
Which is more than enough time to go directly to waypoint 5, but
Yeah.
we can take a
I think
look.
that whole plateau from each end to that contour is what, 200 meters?
And we're just about 20
other way,
meters
the other way.
below the summit. Excuse me.
160.
The summit
Yeah,
of the
that sounds
mound
good.
that we've been
Sounds like a good
crossing
plan.
since the
Could we
beginning of the
fix
dive.
that
We
light,
began
please?
at a depth of about